Hello students, welcome to Paper Wizard. Hope you're all doing well. Today, we're going to discuss the GCE A-Level Biology 2023-2024 MCQ paper. We'll go through each question step by step, breaking down the answers and explanations. So grab a seat, get your notebook ready, and let's dive in. First question. A feature common to lysosomes and peroxisomes is that they are single membrane bounded vesicles. Transport residual materials by exocytosis. Contain oxidizing enzymes that catalyze the breakdown of nucleic acids. Are important in photorespiration. Digest worn-out organelles. Now let's carefully go through each option to figure out which one is correct. The correct answer is option 1. Are single membrane bounded vesicles. Let's break it down. Lysosomes are organelles that contain digestive enzymes. They are responsible for breaking down waste materials and cellular debris. Both of these functions happen inside a structure that is enclosed by a single membrane. Similarly, peroxisomes are also enclosed by a single membrane. They contain enzymes that help break down fatty acids and detoxify harmful substances like hydrogen peroxide. So, both lysosomes and peroxisomes share the characteristic of being single membrane bounded vesicles. Now, let's explore why the other options are incorrect. Option 2. Transport residual materials by exocytosis. While lysosomes may occasionally fuse with the cell membrane to expel waste products after digestion, this is not a key feature of peroxisomes. Peroxisomes focus on breaking down fatty acids and detoxifying substances, not on waste removal via exocytosis. Option 3. Contain oxidizing enzymes that catalyze the breakdown of nucleic acids. This one is tricky because peroxisomes do indeed contain oxidizing enzymes, but they don't break down nucleic acids. Instead, they detoxify harmful compounds like hydrogen peroxide. Lysosomes, on the other hand, contain hydrolytic enzymes which digest proteins, lipids, and carbohydrates, not nucleic acids. Option 4. Are important in photorespiration. While it's true that peroxisomes are involved in photorespiration in plant cells, lysosomes have no role in this process. Photorespiration is specific to plant cells, and lysosomes do not participate in this or any other processes related to plant metabolism. Instead, lysosomes are responsible for the degradation and recycling of cellular waste in animal cells. Option 5. Digest worn-out organelles. This function is specific to lysosomes, which digest cellular debris and damaged organelles through a process called autophagy. Peroxisomes, on the other hand, don't have this function. So, the correct answer remains option 1. Both lysosomes and peroxisomes are single membrane bounded vesicles. So, this wraps up our breakdown of question 1. I hope that clears up any confusion. Let's move on to the next question. Question 2 reads, 2 characteristics that can be seen only in living organisms are adaptation and growth, movement and irritability, change with time and development, metabolism and heredity, synthesis and decomposition. Now, let's work through each option to find the correct answer. The correct answer is option 4, metabolism and heredity. Let me explain why. Metabolism is the sum of all chemical reactions that occur within a living organism, allowing it to maintain life. These reactions include breaking down nutrients to release energy, building up cellular structures, and eliminating waste products. Non-living things do not have metabolic pathways, making this a unique feature of living organisms. Heredity refers to the passing of genetic information from one generation to the next. Through processes like DNA replication and reproduction, living organisms can pass on traits to their offspring. This ability to inherit and pass on genetic material is exclusive to living organisms. So, both metabolism and heredity are core processes found only in living beings. And that's why this option is correct. Now let's break down why the other answers are wrong. Option 1. Adaptation and growth. While living organisms do adapt and grow, adaptation can also occur in non-living things. For example, metal can expand in response to heat, which is a form of adaptation, but it's not a biological process. Growth is also seen in non-living things under certain physical or chemical conditions. For example, crystals grow in size through a process known as crystallization, which occurs when molecules or atoms arrange themselves in a structured, repeating pattern. Option 2. Movement and irritability. Movement is not unique to living organisms. Non-living things, like leaves or dust, can move when external forces act on them, such as wind or water. Irritability, or responding to stimuli, is more exclusive to living organisms. But since movement isn't, this option is incorrect. Option 3. Change with time and development. Change with time happens in non-living things as well. For example, rocks can weather and erode, and metals can rust. Development, on the other hand, refers to biological changes like growth and maturation. But the phrase change with time makes this option incorrect because change can also occur in non-living objects. Option 5. Synthesis and decomposition. Synthesis and decomposition are processes that occur not just in living organisms but also in non-living things. For instance, scientists can synthesize compounds in a lab, and chemical decomposition happens to non-living things like rocks and chemicals. In summary, the only two characteristics exclusive to living organisms are metabolism and heredity. These processes are fundamental to life and do not occur in non-living things. Now let's move on to the third question. Question 3. The question reads, which of the following statements is correct regarding the transmission electron microscope? Specimens are magnified 5 million times. Less electrons may get displayed in cellular structures with dense staining. Living specimens cannot be observed. Three-dimensional appearance of the specimens can be observed. Specimens scatter many electrons, whilst others are absorbed. Let's go through each option to find the right answer. The correct answer is option 3. Living specimens cannot be observed. Let me explain why. A transmission electron microscope, TEM, uses a beam of electrons to pass through very thin slices of specimens. For this to work, the specimen must be treated with chemicals, fixed, and dehydrated, which unfortunately kills the specimen in the process. This means you can't observe living specimens in a TEM. However, this process provides high-resolution images of the internal structure of cells, making TEM a valuable tool in cellular biology. Now, let's break down why the other answers are wrong. Option 1. Specimens are magnified 5 times 10 to the power of 6 times. TEMs do provide high magnification, but the maximum magnification typically ranges between 1 to 2 million times, not as high as 5 million. So, this option exaggerates the magnification power of the TEM. Option 2. Less electrons may get displayed in cellular structures with dense staining. In a TEM, the denser areas of a specimen absorb more electrons, which results in these regions appearing darker in the final image. This happens because fewer electrons are transmitted through the denser parts of the specimen, not because fewer electrons are displayed. The electrons that do pass through the less dense areas will hit the detector, making those regions appear lighter. So the statement, less electrons may get displayed in cellular structures with dense staining, is incorrectly phrased. The correct understanding is that fewer electrons pass through the denser regions, which leads to them appearing darker in the image, while the less dense regions appear lighter because more electrons pass through. Option 4. Three-dimensional appearance of the specimens can be observed. TEM produces two-dimensional 2D images because the electrons pass through a thin slice of the specimen. If you want a three-dimensional 3D view, you would need a scanning electron microscope, SEM, which scans the surface of the specimen to create a 3D image. So, TEM is not capable of 3D imaging. Option 5. Specimens scatter many electrons whilst others are absorbed. While some scattering of electrons does occur in TEM, the technique primarily focuses on transmission, meaning electrons pass through the specimen to form an image. Electron scattering is more of a feature of SEM, where the surface of the specimen is scanned and scattered electrons are collected. So, this statement is more relevant to SEM than TEM. To sum it up, TEM provides high-resolution 2D images of the internal structures of non-living specimens. And this is why option 3 is the correct answer. Living specimens cannot be observed. Now let's look at question 4. The question asks, select the response which correctly indicates the event and phase in the eukaryotic cell cycle. DNA replication. G0 phase, synthesis of proteins. G1 phase, chromatin formation. G2 phase, production of cellular organelles. S phase, duplication of centrosome. M phase. Let's go through each option to find the correct answer. The correct answer is option 2. Synthesis of proteins, G1 phase. Here's why. The G1 phase, first gap phase, occurs right after mitosis and before the S phase, when DNA is replicated. During G1, the cell grows and synthesizes various proteins, including enzymes required for the upcoming DNA replication in the S phase. This is when the cell prepares itself for DNA synthesis, making it the most appropriate phase for protein synthesis. Now, let's look at why the other options are wrong. Option 1. DNA replication, G0 phase. This is incorrect because DNA replication takes place during the S phase, not in the G0 phase. The G0 phase is a resting phase, where cells exit the cell cycle and are not actively preparing for division. Cells in G0 typically carry out regular metabolic functions but do not replicate their DNA. Option 3. Chromatin formation, G2 phase. Chromatin formation refers to the packaging of DNA into chromatin, which occurs primarily after DNA replication during the S phase. The G2 phase is mainly responsible for the final preparations for mitosis, including additional cell growth and the production of organelles and proteins necessary for cell division. Additionally, the cell checks for any errors in DNA replication and, if needed, repair mechanisms are activated to ensure the accuracy of the genetic material before the cell proceeds to mitosis. So, option 3 is incorrect. Option 4. Production of cellular organelles, S phase. The S phase is specifically involved in DNA replication, not the production of cellular organelles. Organelles are mainly produced during the G1 and G2 phases, as the cell grows and prepares for division. This means that cellular organelle production doesn't occur in the S phase. Option 5. Duplication of centrosome, M phase. Centrosome duplication begins in the S phase and is completed during the G2 phase. The M phase, mitosis, is focused on dividing the already duplicated chromosomes and cellular contents between the two daughter cells, so centrosome duplication doesn't occur in the M phase. To wrap it up, the G1 phase is when the cell synthesizes the proteins necessary for cell growth and DNA replication, making option 2 the correct answer. Alright, let's tackle question 5. The question reads, in allosteric regulation of enzymes, regulatory molecules bind reversibly to the active site of the enzyme. Regulatory molecules bind to the enzyme via non-covalent interactions. An activator molecule that binds to a particular subunit will affect the active site of that subunit only. Inhibitory molecules affect the function of the enzyme, but not the shape. ATP functions as an allosteric activator. Now let's analyze the options carefully. The correct answer is option 2. Regulatory molecules bind to the enzyme via non-covalent interactions. Let me explain why. Allosteric regulation occurs when a regulatory molecule, which can be either an activator or inhibitor, binds to an enzyme at a site distinct from the active site, known as the allosteric site. This binding typically occurs through non-covalent interactions such as hydrogen bonds, ionic bonds, or hydrophobic interactions. These non-covalent interactions allow the binding to be reversible, meaning that the regulatory molecule can detach when it's no longer needed. This allosteric interaction leads to a conformational change in the enzyme's structure, which can either enhance, if it's an activator, or reduce, if it's an inhibitor, the enzyme's activity. Now, let's discuss why the other options are incorrect. Option 1. Regulatory molecules bind reversibly to the active site of the enzyme. This option is incorrect because, in allosteric regulation, the regulatory molecules do not bind to the active site. Instead, they bind to the allosteric site, which induces changes in the enzyme's activity. If a molecule binds to the active site, it would be classified as competitive inhibition, which is different from allosteric regulation. Option 3. An activator molecule that binds to a particular subunit will affect the active site of that subunit only. This statement is incorrect because when an activator or inhibitor binds to the allosteric site, it causes a conformational or structural change in the entire enzyme, not just the subunit where the binding occurred. Many enzymes are made up of multiple subunits, and these subunits often work together to perform the enzyme's function. In these cases, when a regulatory molecule binds to one subunit, it doesn't just affect that subunit alone. The binding event induces a global change in the enzyme structure, affecting all subunits and their active sites. This means that a change at one site can influence the enzyme's overall activity, potentially altering the function of other subunits as well. This process is essential for the coordination of the enzyme's activity, particularly in enzymes that work in concert, like hemoglobin. Option 4. Inhibitory molecules affect the function of the enzyme but not the shape. This is wrong because in allosteric regulation, inhibitors change the shape of the enzyme when they bind to the allosteric site. This conformational change affects the enzyme's function, either slowing it down or stopping it altogether. So, inhibitors definitely influence the enzyme structure as well as its activity. Option 5. ATP functions as an allosteric activator. This statement is misleading because the role of ATP as an allosteric regulator varies depending on the enzyme and the specific metabolic context. For example, in the glycolytic pathway, ATP acts as an allosteric inhibitor of the enzyme phosphofructokinase 1, or PFK1, which controls the rate of glycolysis. When ATP levels are high, it binds to the allosteric site of PFK1, reducing the enzyme's activity and slowing down ATP production, serving as a negative feedback mechanism to prevent excess ATP generation when energy is abundant. On the other hand, ATP can act as an allosteric activator in other pathways. For instance, ATP activates glutamine synthetase, an enzyme involved in nitrogen metabolism, enhancing its activity when ATP is plentiful to support biosynthetic processes. Thus, the statement is misleading because it suggests that ATP always acts as an activator. In reality, ATP can act as either an activator or inhibitor depending on the enzyme and the metabolic needs of the cell. So, to sum it up, option 2 is the correct answer because regulatory molecules bind via non-covalent interactions to the allosteric site of the enzyme, influencing its activity in a reversible manner. Alright, now we're on to question 6. The question reads, in ethyl alcohol fermentation, one molecule of glucose produces one molecule of pyruvate and two molecules of NADH. Pyruvate is reduced directly to ethanol using NADH. One molecule of CO2 is produced from one molecule of glucose. The final hydrogen acceptor is an inorganic compound. Two molecules of ATP are produced from one molecule of glucose. Let's analyze these options and figure out the correct answer. The correct answer is option 5. Two molecules of ATP are produced from one molecule of glucose. Let me explain why. In ethyl alcohol, ethanol fermentation, the process begins with glycolysis. During glycolysis, one molecule of glucose is broken down into two molecules of pyruvate, which generates two molecules of ATP and two molecules of NADH. Following glycolysis, pyruvate is reduced to ethanol through a two-step reaction. In this process, NADH is oxidized back to NAD+, allowing glycolysis to continue in the absence of oxygen. However, no additional ATP is produced during the fermentation steps. So, in total, we have two ATP molecules produced from the breakdown of one molecule of glucose. Now, let's explore why the other options are incorrect. Option 1. One molecule of glucose produces one molecule of pyruvate and two molecules of NADH. This is incorrect because one molecule of glucose actually produces two molecules of pyruvate during glycolysis. Along with this, two molecules of NADH are generated. So it's not just one pyruvate. Option 2. Pyruvate is reduced directly to ethanol using NADH. This is wrong because the conversion of pyruvate to ethanol is a two-step process. First, pyruvate is decarboxylated to form acetaldehyde, and only then is acetaldehyde reduced to ethanol using NADH. So, pyruvate is not directly reduced to ethanol in a single step. Option 3. One molecule of CO2 is produced from one molecule of glucose. This is also incorrect. During ethanol fermentation, two molecules of CO2 are produced from one molecule of glucose. Here's why. For every molecule of glucose, two molecules of pyruvate are formed, and each pyruvate undergoes decarboxylation, producing one molecule of CO2 per pyruvate. Therefore, two CO2 molecules are released from one glucose molecule. Option 4. The final hydrogen acceptor is an inorganic compound. This statement is wrong because in ethanol fermentation, the final electron, hydrogen acceptor, is an organic compound, specifically acetaldehyde, which is reduced to ethanol. In contrast, in aerobic respiration, an inorganic compound like oxygen acts as the final electron acceptor. But in this case, the process is anaerobic, so the acceptor is organic. In summary, the entire process of ethyl alcohol fermentation yields a net gain of two ATP molecules from one molecule of glucose, making option 5 the correct choice. Alright, we've made it to question 7. The question reads, which of the following statements regarding glycolysis of one molecule of glucose is correct? There is a net yield of 4 ATP molecules. 2 hydrogen ions are released. It partially depends on molecular oxygen. 2 NADH molecules are formed. Part of glycolysis takes place in the outer membrane of mitochondria. Let's break this down to find the correct answer. The correct answer is option 4. 2 NADH molecules are formed. Let me explain why. Glycolysis is a critical metabolic pathway that occurs in the cytoplasm of the cell, where one molecule of glucose, a 6-carbon sugar, is broken down into 2 molecules of pyruvate, each containing 3 carbons. This pathway consists of several enzymatic steps, which involve energy investment and energy generation phases. In the initial steps of glycolysis, glucose undergoes a series of transformations and is eventually split into 2 molecules of glyceraldehyde 3-phosphate, G3P. This occurs after an energy investment phase where 2 ATP molecules are consumed to prepare the glucose for splitting. At this point, we have 2 molecules of G3P, each of which will undergo the same subsequent reactions to eventually form pyruvate. The next crucial step for producing NADH occurs when each molecule of G3P is oxidized into 1,3-bisphosphoglycerate. This oxidation step is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. During this reaction, NAD+ acts as an electron acceptor. As G3P is oxidized, high-energy electrons and protons, H+, are transferred to NAD+, reducing it to NADH. For each molecule of G3P, one NAD+ is reduced to NADH. Since there are 2 G3P molecules, one from each half of the split glucose, this results in the production of 2 NADH molecules overall from one molecule of glucose. In simpler terms, each G3P molecule generates 1 NADH, and since 2 G3P molecules are processed, 2 NADH molecules are produced. These NADH molecules act as carriers of high-energy electrons and protons, which are later used in oxidative phosphorylation in the mitochondria during aerobic respiration to generate more ATP. Now let's explore why the other options are incorrect. Option 1. There is a net yield of 4 ATP molecules. This is incorrect because although 4 ATP molecules are produced during glycolysis, 2 of them are consumed in the early steps of the process. So, the net yield is only 2 ATP molecules, not 4. Option 2. 2 hydrogen ions are released. Gete. This statement is false because more than 2 hydrogen ions are released during the process. During glycolysis, when glyceraldehyde 3-phosphate, G3P, is oxidized to 1,3-bisphosphoglycerate, 2 NADH molecules are produced, and with them, 2 hydrogen ions, H+, are released into the cytoplasm. Additionally, 2 more H+ ions are released in other steps of glycolysis, bringing the total number of hydrogen ions released to 4 per molecule of glucose. Therefore, the correct number of hydrogen ions released during glycolysis is 4, not 2, making the statement incorrect. Option 3. It partially depends on molecular oxygen. This is incorrect because glycolysis is an anaerobic process, meaning it does not require oxygen to occur. Whether oxygen is present or not, glycolysis can proceed in the cytoplasm. Oxygen comes into play during the later stages of cellular respiration, like oxidative phosphorylation, but it has no role in glycolysis. Option 5. Part of glycolysis takes place in the outer membrane of mitochondria. This is also incorrect. Glycolysis happens entirely in the cytoplasm of the cell, not in the mitochondria. The mitochondria are involved in the citric acid cycle and oxidative phosphorylation, but glycolysis occurs outside of the mitochondria, in the cytoplasm. So, to wrap this up, during glycolysis, 2 NADH molecules are produced, which makes option 4 the correct answer. Now, let's look at question 8. The question gives us a series of events that took place during the evolution of organisms, which are A. Saturation of water bodies with oxygen. B. Oxidation of Fe2+. C. Increase in photosynthetic bacterial populations. D. Origin of cyanobacteria. And asks, what is the correct sequence of these events? The correct answer is option 5. D, B, A, and C. Here's why. D. The origin of cyanobacteria. Cyanobacteria are some of the earliest photosynthetic organisms that appeared on Earth. They were able to produce oxygen through photosynthesis, and this process would go on to transform the Earth's atmosphere. The presence of cyanobacteria marks the beginning of oxygen production. B. Oxidation of Fe2+. As cyanobacteria began producing oxygen, the oxygen reacted with Fe2+, ferrous ion, present in the oceans. This oxidation turned Fe2+ into Fe3+, ferric ion, leading to the production of banded iron formations that we can still see in rocks today. A. Saturation of water bodies with oxygen. After the iron in the oceans was oxidized, oxygen levels continued to rise. Eventually, the water became saturated with oxygen, which then began to accumulate in the atmosphere. C. Increase in photosynthetic bacterial populations. With oxygen levels increasing, this created an environment that allowed more oxygen-producing photosynthetic bacteria to evolve and thrive, increasing the population of these bacteria. Why the other answers are incorrect? Option 1, A, B, C, and D. This option is incorrect because it places saturation of water bodies with oxygen before the origin of cyanobacteria. Cyanobacteria were responsible for producing the oxygen in the first place, so they had to appear first. Option 2, C, A, B, and D. This is incorrect because it places the increase in photosynthetic bacterial populations before the oxidation of Fe2+. However, the rise in oxygen from cyanobacteria is what caused the oxidation of iron, so this sequence is incorrect. Option 3, C, B, A, and D. Similar to the previous option, this one also places the increase in photosynthetic bacterial populations too early, before the origin of cyanobacteria, which is not correct. Option 4, D, A, B, and C. This sequence is close but incorrect because it places the saturation of water bodies with oxygen before the oxidation of Fe2+, when in fact the oxidation of Fe2+ had to occur first, as oxygen initially reacted with iron in the oceans. To summarize, the correct sequence of events is D. Origin of cyanobacteria. B. Oxidation of Fe2+. A. Saturation of water bodies with oxygen. And C. Increase in photosynthetic bacterial populations. This aligns with what's known as the Great Oxygenation Event, a major turning point in Earth's history. All right, we're on to question 9. The question reads, which of the following pairs of organisms have the highest number of common characteristics? 1. Bat and crow. 2. Lizard and turtle. 3. Ichthyophis and Taenia. 4. Alva and Pogonatum. 1. Pinus and Cycas. Let's go through each pair and figure out which has the most common characteristics. The correct answer is option 2, lizard and turtle. Here's why. Lizards and turtles both belong to the class Reptilia, meaning they share many fundamental characteristics associated with reptiles. These shared traits include: both lizards and turtles are ectothermic or cold-blooded, meaning they rely on external sources of heat to regulate their body temperature, a hallmark of reptiles. Both animals have scales covering their skin, which protect their bodies from dehydration and injury. Scales are a distinctive feature of reptiles. Both lizards and turtles lay eggs with leathery, flexible shells, unlike the hard-shelled eggs of birds. This feature is crucial for terrestrial reproduction in reptiles, as the leathery shell prevents water loss while still allowing gas exchange. Also, both reptiles have lungs for respiration and a 3-chambered heart. Their circulatory systems allow for partial mixing of oxygenated and deoxygenated blood, which is typical in most reptiles. An exception to this is crocodiles, which have a 4-chambered heart, allowing for a complete separation of oxygenated and deoxygenated blood, similar to birds and mammals. These shared characteristics illustrate why lizards and turtles have the highest number of common features among the given options. Now, let's break down why the other options are incorrect. Option 1. Bat and crow. While both bats and crows are flying vertebrates, they belong to different classes within the animal kingdom. Bats are mammals, classified under class Mammalia, whereas crows are birds, belonging to class Aves. Bats give birth to live young, viviparous, while crows lay eggs, oviparous. This fundamental difference in reproductive strategy reflects their different evolutionary paths. Bats have fur, a characteristic of mammals, while crows are covered in feathers, a defining trait of birds. Feathers are specialized for flight and insulation, whereas fur serves as insulation for mammals. Due to these significant differences in physiology and classification, bats and crows share fewer common characteristics compared to reptiles like lizards and turtles. Option 3. Ichthyophis and Taenia. Ichthyophis is a genus of caecilian, a type of amphibian. Amphibians are vertebrates that generally live part of their life in water and part on land, and they have smooth, permeable skin that requires moisture. Taenia, on the other hand, is a genus of parasitic flatworms, tapeworms, which belong to the phylum Platyhelminthes. These organisms are invertebrates, meaning they lack a backbone, and they have a completely different body structure and life cycle from amphibians. Key differences include: Ichthyophis, as an amphibian, has a vertebrate body with limbs, or limbless in some cases like caecilians, and a complex organ system. Taenia, being a flatworm, has a very simple flattened body without limbs or a complex organ system. Ichthyophis undergoes a life cycle that involves both aquatic and terrestrial stages, while Taenia has a parasitic life cycle involving hosts such as humans or animals. They are vastly different in their reproductive strategies and development. Thus, these two organisms have minimal similarities, making them far less related compared to lizards and turtles. Option 4. Alva and Pogonatum. Alva is a genus of green algae, phylum Chlorophyta, which are simple, aquatic organisms that perform photosynthesis. They are primarily found in marine environments and lack complex structures like stems, roots, and leaves. Pogonatum, on the other hand, is a genus of moss, phylum Bryophyta, which are terrestrial plants that also perform photosynthesis but have a more complex structure compared to algae. Mosses are non-vascular plants, meaning they do not have specialized tissues like xylem and phloem for water and nutrient transport like higher plants do. Key differences include: Alga is a simple, sheet-like alga with minimal differentiation in structure, while Pogonatum has more developed plant-like features such as rhizoids, root-like structures, stems, and leaf-like structures. Alga typically inhabits aquatic environments, whereas Pogonatum is found in moist terrestrial habitats. Although both are photosynthetic organisms, they belong to different evolutionary lineages and phyla, making them quite distinct from each other. Option 5. Pinus and Cycas. Both Pinus and Cycas belong to the group of plants known as gymnosperms, plants that produce seeds without flowers, but they represent different subgroups within gymnosperms. Pinus is a conifer, part of the Coniferophyta group, which includes trees like pines, firs, and spruces. These plants have needle-like leaves and produce seeds in cones. Cycas, on the other hand, belongs to the Cycadophyta, a group of ancient plants with a palm-like appearance and large compound leaves. Cycads reproduce using large seeds and cones, but their reproductive structures and growth patterns differ significantly from conifers. Key differences include: while both produce cones, their structures are quite different. Pinus produces both male and female cones on the same plant, while Cycas are often dioecious, which means they have separate male and female plants. Cycads are considered more primitive than conifers and have a more ancient evolutionary lineage, with some species being described as living fossils. Although both are gymnosperms, their differences in reproductive biology and structure are more pronounced compared to the similarities between reptiles like lizards and turtles. To sum up, lizards and turtles share the most common characteristics as they both belong to the Reptilia class, making option 2 the correct answer. Alright, now we've arrived at question 10. The question asks, which of the following are unique characteristics of some phyla of the kingdom Animalia? A. Internal fertilization. B. Parapodia. C. Radula. D. Nephridia. Let's go through each of these characteristics and see which ones are truly unique to specific phyla. The correct answer is option 3, B and C only. Let's break down why this is the correct answer. B. Parapodia are lateral appendages found in some members of the Annelida phylum, particularly polychaetes. Parapodia are used for locomotion and gas exchange, and they are a distinctive feature of this group, making them a unique characteristic. C. Radula is a specialized feeding organ found in the mollusca phylum, particularly in gastropods like snails. It is used to scrape or cut food before ingestion. This is a unique and defining feature of mollusks. So, Parapodia and Radula are both distinctive characteristics associated with specific phyla, making option 3 the correct choice. Now let's see why other answers are incorrect. Option 1, A and C only. This is incorrect because A. Internal fertilization occurs in a variety of animal groups, such as mammals, reptiles, and some fish. It is not unique to any particular phylum in the kingdom Animalia, which makes it a general characteristic rather than a unique one. Option 2, A and D only. As mentioned in the previous answer, internal fertilization is not unique to any particular phylum in the kingdom Animalia. And while D, Nephridia, are excretory structures found in annelids and some other invertebrates, they are not as distinct to one group as Parapodia and Radula are. Option 4, B and D only. Parapodia are unique to some annelids, but Nephridia are present in various invertebrate phyla. Therefore, this combination doesn't hold as much uniqueness as Parapodia and Radula. Option 5, C and D only. While Radula is unique to mollusca, Nephridia are not unique enough to any one phylum. This makes this option less correct compared to B and C. In summary, option 3 is correct because Parapodia and Radula are truly unique to their respective phyla, making them distinctive features of the animal kingdom. And that wraps up question 10. I hope the explanations were clear and helpful. This is the end of today's video. Thank you so much for watching. Stay tuned for the next session where we'll continue discussing the remaining questions of this paper. Until then, keep studying and try the remaining questions on your own. If you learned something new today, don't forget to give a thumbs up, subscribe, and click on the bell icon so you'll be notified when the next video is ready. Also, comment your thoughts below and share this video with your friends. Until we meet again, take care.